Hello, everybody. Welcome to the third lecture in Chapter 10. So this is a much shorter chapter. We'll be looking at two difficult to see aspects of muscle functioning, still somewhat important, as you'll see. And towards the end of this lecture, we'll say a few words, very few words about cardiac and smooth muscle to round out Chapter 10. In addition to this recording, you should be seeing a recording of Chapter 11, which is very, very short. And I hope to see all of you on Tuesday, ready to move on to the nervous system. If you have any questions about this recording or the Chapter 11 recording, please send me a message or bring your questions to lecture and lab on Tuesday. So let me start this screen sharing and we'll move on with the slides. Okay, let's make this full screen. I'm going to minimize this window here. So what we have looked at in this chapter is how skeletal muscles contract. We looked at the necessary structures in the sarcomere. In the second lecture, we looked at how our nervous system starts the contraction and how the muscle cell responds by opening sodium channels first and then releasing calcium from the sarcoplasmic reticulum, and then that calcium changing the configuration of the thin filament to allow the thick filaments to grab onto those actin proteins of the thin filament and start dragging them towards the center of the sarcomere. And this is all fine. However, there is an aspect to our muscle force, namely the fact that we can regulate how much force we create from a single muscle. So imagine this scenario. You are reaching into the fridge, into the egg crate, and picking out an egg very carefully. The same muscles that bend your fingers ever so lightly to pick out this egg from an egg crate can also force your fingers to crush that eggshell. So how is it possible that in 1 minute we very gently pick out an egg, and the next second we can instruct our muscles to create more force and crush this egg? We're not growing new muscles in that second. We are not adding any other body parts. So how can we take a muscle and instruct it to create less or more force? And a very intuitive way of doing this is happening through this motor unit recruitment action. So let's take a look at our skeletal muscle, and our poster child will be here, biceps brachii. So I mentioned this before in lecture that our biceps brachii have roughly 200,000 cells. Those 200,000 cells are divided into functioning unit, the motor units. 2,000 cells form a single motor unit in our biceps brachii. So it means that roughly there are 100 motor units. And the key to increasing or decreasing muscle force in the biceps brachii and all other muscles is that these motor units have independent nerve supply. So on this image, you see a motor unit made up of two muscle cells. And here you see a green motor unit where the green axons contact three cells. So these two motor units are acting independently. They can be turned on and off independent of each other. So think about this biceps brachii or any other muscle. If you turn on one motor unit out of 1,000 or out of 100, we create a fraction of the total force. If we turn on half the motor units, we are generating a lot more force. And if we turn on, activate all the motor units in a muscle, we create the maximal force. So motor unit recruitment can add to the muscle force by activating 1, 5, 10, 50%, and if needed, 100% of the motor units, activating all muscle cells to create a maximal force. So this motor unit recruitment is one way to create a little or large force from the very same muscle. Little force, we activate a few motor units. Large force, we activate all motor units in this particular muscle. So I'm going to move over here. There is another way of changing muscle force, and this is going to be even more puzzling because a single motor unit can be regulated to create a little or large force. Now, how is that possible? We turn on the motor unit, all muscle cells in the motor unit at the same time. How could we possibly change the force from a single motor unit? So let's take a look at first what a motor unit does in terms of force creation. So when nerve impulse is delivered to a motor unit, we have this latent period for a few milliseconds, followed by a contraction period, and then there's a relaxation period. Let's take a look at what is happening in this twitch contraction. So the latent period is taken up by calcium being released from sarcoplasmic reticulum during the contraction. This is where the thick filaments grab onto the thin filaments and pull them towards the middle of the sarcomere. And once we stop signaling the muscle or the motor unit to contract, the relaxation period comes. This is the time when calcium is vacuumed out from the sarcomere and contraction stops. And so muscle force goes back to the original zero muscle force. So here in this twitch contraction is the secret to how we can increase muscle force from a single motor unit. So let's take a look at this. So here's again our twitch contraction. Single motor unit received one nerve impulse. But take a look at what happens when we deliver two nerve impulses. If the second nerve impulse is delivered to the single motor unit just at the beginning of the relaxation period, the next contraction of this motor unit will produce higher force. Not only that, take a look at when we deliver four of these, one after the other, four nerve impulses. The delivery has to happen in the beginning of relaxation period, but if we time these nerve impulses right, we can keep increasing the force created by a single motor unit. And here's the maximum to the right when we time many nerve impulses in a way that we maximize muscle force from a single motor unit. So how is this possible? And the explanation is in the level of calcium. During the first contraction, maybe only 20% of the total calcium is released from the sarcoplasmic reticulum. But when we add the second nerve impulse, another 20% is released. More calcium means more sarcomeres will be contracting. And with several nerve impulses timed one after another, we are releasing more and even more calcium from sarcoplasmic reticulum. And when we reach this fused tetanus state, 100% of the calcium is now released. There's no further increase. We plateau here we max out a muscle contraction. So this way of increasing muscle force from a single motor unit is called wave summation. In a living muscle, motor unit recruitment and wave summation are combined to give a very smooth increase of muscle force. Think about this scenario. You are exiting the class and trying to open the door. If the door is a little stuck, a gentle pull is not going to open the door, and you will gradually increase that force until the door swings open. That gradually increasing force to open the door is coming from a combination of wave summation. Individual motor units are stepping up their force, but you are also recruiting more motor units to create a larger force to open the door. So these two waves, motor unit recruitment and wave summation, are the two mechanisms how we can create less or more force from a single muscle. And so this is the explanation linking this to calcium levels. And this is motor unit recruitment. Let's say a few words about this muscle tone. This is yet another dark secret of muscles, that in a totally seemingly relaxed muscle, there are a couple of motor units that are turned on periodically, and this causes what's called a muscle tone. If you touch your skeletal muscles, when you squeeze on them with your fingers, they resist the squeeze. They are firm to the touch. If we would completely relax this muscle, let's say cut all nerve impulses to this muscle, this muscle would feel spongy. You could squeeze it much easier. This is called flaccid muscle. And a flaccid muscle just means that all nerve impulses lost to that muscle. Usually it's a sign of damage to the neuromuscular junction, either chemical or physical damage, or physical damage to the neurons that would be bringing nerve impulses to the muscle. And the last topic here is somewhat related to physiology. This is a neat way our body is programmed, because we cannot really change this. But as muscles develop during embryonic and fetal development, some motor units will develop as slow oxidative. We'll just look at the two extremes here. And the other extreme is fast glycolytic fiber. So what does this mean? This is related to ATP production. So the slow oxidative fibers have lots of myoglobin, have lots of mitochondria, and oxidative means that they rely on ATP produced primarily by oxygen-consuming mitochondria. Because they are loaded with myoglobin, they appear as dark, stained muscle cells under the microscope. The extreme here, fast glycolytic, they have few mitochondria, and they derive a lot of their ATP from the anaerobic respiration. And these muscles tend to have less myoglobin. Take a look at the fast glycolytic muscle, much paler, much lighter color because of low myoglobin. The utility of these muscles is that slow oxidative fibers, muscle cells, you find in muscles that are almost constantly active. These are the muscles that maintain posture, whether it's posture of the torso, maintain balance by coordinating thigh, leg, and foot movement. And the fast glycolytic fibers are at higher proportion in muscles where we need only periodic high burst of force. And this is mostly shoulder and arm muscles. So the culinary aspect of all this is that when you cook muscles that are rich in slow oxidative fibers, they turn dark because of the high myoglobin content. We call this dark meat. And what's a dark meat on a chicken or a turkey? It's the thigh drumsticks. What is white meat? Rich in fast glycolytic fibers. It's the wings, the pectoralis major, which is the breast meat on a chicken or turkey. Those are lighter colored muscles, even after we cook or bake them, because they have low myoglobin content. So after this conclusion to the skeletal muscles, let's say a few words about cardiac muscle. Excuse me. So cardiac muscles differ from skeletal muscles by having intercalated discs, desmosomes, and gap junctions. The flow of the flow of calcium into cardiac muscle is coming from the outside because the sarcoplasmic reticulum is not well developed in cardiac muscle. Contraction time, the twitch contraction is much, much longer. We'll emphasize this a lot in the heart chapter in AMP2. We also emphasize this one that cardiac muscle cells contract after they are stimulated by the internal pacemaker. There are nerve impulse generating fibers inside the heart. And again, we'll detail this in AMP2. I'm not going to say much about this slide because we have seen something very similar in the tissue chapter, illustrating that the cardiac muscle cells are branched. They have a single nucleus, and we have sarcomeres arranged here, very similar to skeletal muscles. With the smooth muscle, we'll say a few things. Smooth muscles bundle together, group together in two different ways. One way where the smooth muscles pass on information among each other, a single nerve impulse can activate many smooth muscle cells. This happens because gap junctions relay information between smooth muscle cells. They can be autorhythmic, really typical in the digestive system, stomach, small intestine, large intestine. The multi-unit smooth muscle is where each cell receives its own nerve impulse. Arteries, large arteries have multi-unit smooth muscle, large airways, and our favorite erector pili muscle. This ciliary body and iris are smooth muscles located inside the eyeballs. We'll detail those in AMP2. So because smooth muscles are non-striated, they do not have classical sarcomeres. This is why we look at, just very briefly, how contraction is possible without sarcomeres. Well, as it turns out, smooth muscle cells do have thick and thin filaments. But these thick and thin filaments are arranged differently, kind of scattered fashion. And the thin filaments are anchored in these dense bodies, these sphere-looking spots on the cell surface. And when contraction happens, these dense bodies are pulled closer to each other, and the entire cell shortens as a consequence of that. So it looks very different from how skeletal and cardiac muscle cells contract. We do have thick and thin filaments, but we have no Z-lines or Z-disc. They are replaced by these dense bodies on the muscle cell surface. And let's say a few words about the physiology. So sarcoplasmic reticulum is poorly developed in smooth muscle cells. We do have a very different protein, calmodulin, to regulate turning on and off smooth muscles. And the smooth muscles have an extreme large ability to stretch. In previous lectures, I may have mentioned the uterine smooth muscles that have to stretch from a small fist size to the size of a fully grown fetus by 9 months. And this is a unique property of smooth muscle cells. Smooth muscle cells are also very much used to create constant force. Maintaining blood pressure is probably the best example. Inside our blood vessel walls, there's a layer of smooth muscles, and they need to contract basically throughout our lifetime to maintain blood pressure. And let me stop screen sharing here. This is the end to the third part of the lecture in chapter 10. Again, you'll find this recording together with chapter 11 recording. This will be an older recording in your class shell. I'll send a message out to everybody. So thank you for paying attention to this, and I'll see you in lecture next Tuesday.